Good afternoon, friends. This is the second day of Drug Discovery Hackathon training program, and we welcome all of you and take the advantage of the lectures that are being given in this training program. And the lecture today is by Professor Prasad V. Bharatam from Mohali. He is one of the most well-known medicinal chemists and computational chemists of the country, who also has played a very important role in establishing pharmacoinformatics for the first time in Mohali and guided a large number of PhD students and MPharm and other postgraduate, graduate and postdoctoral scientists. While he was trained as a computational chemist, he worked on a range of problems. When he was a postdoc, he also worked on the defense problems of United States, came back to GNDU, and from there he went to Mohali almost 20 years before. And there, as a medicinal chemist, he has converted himself from a computational medicinal chemist to an experimental medicinal chemist, and the molecules that he has designed, he went on to synthesize, and this is something which is very remarkable. And one of the most important features of Professor Prasad's research is he pays a lot of attention on the fundamental aspects and then discovered many new bonding principles at the fundamental level and then deciphered their importance in the mechanism of drug action. And today he will be discussing with you the basics of drug action, and we are extremely lucky that one of the most eminent personality in the area of drug discovery is going to talk to us. On behalf of all of you, I welcome Professor Prasad. Thank you very much, sir, for kindly accepting our invitation and then sparing your valuable time. Now, I request Bharatam to give his lecture on the mechanism of drug action. Bharatam, please. You will have to unmute yourself. We are not. Professor Prasad, you have to unmute yourself.
Yes. Am I audible now?
Now you are audible. Yeah, please go ahead.
Okay. Thank you, Dr. Sastry, for giving this opportunity. It's a nice initiative taken by National Innovation Foundation as well as Prime Minister's Office. Now CSIR NIST is supporting this thoroughly from all possible perspectives. And now I would like to start my presentation with the title. I hope all of you can see it. This is, can I go ahead? Is it visible to all? Dr. Sastry? Okay, I hope it is fine. Now this is my name and my address. You already know me a little bit. I am going to present this in this following way. Let us see, first of all, what are the activities in drug discovery. The very first thing is to understand the problem. This is what Dr. Sastry emphasized in yesterday's his own lecture. He said that understanding the basics is very important. That is why we are going to start with that. Then design the compounds using rational methods. Rational methods are very typical name, rational method. What is it? Rational does not mean it is proven or 100% truthful methods. It means that logically employed methods. That means every scientist uses logic only. In drug discovery, logic is very important. But then what is the meaning of rational drug design? It means that we are using some technologies which are known as rational drug design technologies. Under the heading called pharmacoinformatics, we are teaching in our institute, but in the rest of the world, it is also known as computer-aided drug design methods. Now, once we finish rational drug design, we go to the synthesis of the new species. Once we do that, we go to in vitro testing and then go to in vivo testing. Once we are happy with that, we go to the preclinical pharmaceutics. That is called preclinical formulation and other related things. Then once this is all done, clinical studies are approved, and once we enter into clinical studies, it will take a long time to get out of it. And once we successfully come out, we may have a drug in our hand. Let us go for that. So in this process, very first thing is to understand the basics. Next is to apply the rational drug design methods. We are doing the drug discovery hackathon with the help of the second thing, that is rational drug design methods. However, before going into it, we need to pay attention to understanding the basics of drug action. That is what is the focus of my lecture, and I shall go to this point. We are talking about drug discovery hackathon using informatics methods. Information technology has provided many facilities. 30 years ago, these facilities did not exist. Many facilities started, bioinformatics, chemoinformatics. They are very young topics. These young topics are helping us to go to other informatics fields like biomedical informatics, ADME informatics, toxicity informatics. Likewise, there are so many methods of information technology which can be applied to various subfields of pharmaceutical sciences. That is what is the science-oriented topics. Next is the service-oriented topics. This is the pharmacy informatics, clinical informatics, healthcare informatics, medical informatics, nursing informatics, and things like that. These are all service-oriented sectors which come into the picture after the drug is identified. Drug discovery is done and the service starts. So we are now in the science-oriented topics, that is drug discovery informatics. Now, once we have this, we go ahead and find out specific therapeutic application-oriented topics like cancer or anti-diabetic are now antiviral. COVID, anti-COVID is what is our specific therapeutic application-oriented informatics topic in pharmaceutical sciences. That is what is defining our drug discovery hackathon. Having done this, many topics where nanotechnology, biotechnology, and information technology are integrated with pharmaceutical sciences. Thus, we are going to have a wholesome technology cum science oriented topic. This is a flow chart showing the same what I described right now. Neuroinformatics, genome, biosystems, all of them contribute finally towards pharmacoinformatics, and eventually we design drug. After that, we will have service-oriented pharmacoinformatics. Now let us go to the basics. Basics are known to all of us. If some of the terminology is so primitive that you all get the feeling that we already know this, why we are giving a lecture on this topic today. Yes, it is important to recall all our basics and see how it is, these basics are linked directly to the research that we are going to do in the coming 6 months through the DDH program, the Drug Discovery Hackathon program. Having done that, let us see what are the topics today. Theories of drug action. There are a few theories that are already proposed to understand the drug action. There are some excellent books on this topic. You can refer to those and we can understand the theories of drug action. Then theories of enzymatic action. This is another important topic because enzymes are very important for us. We are going to deal with enzymes, and there is a lot of computational and non-computational theories, fundamentals about the drug action, enzymatic action, as well as the action of the enzymes on the substrate. Those things are discussed in this particular topic called theories of enzymatic action. Now, again, Dr. Monday, Ashade mentioned about the drug receptor interactions. He discussed about the receptors. We are going to touch upon them once again. Inhibitor enzyme interactions. These are slightly different from drug receptor interactions. When drug and receptor interact with each other, there is no chemical reaction which is happening because the interactions are more or less associations. Association dissociations are happening when we talk about drug and receptor interactions. But when we talk about inhibitor and enzyme, actually what is happening is there is an enzyme substrate reaction. That particular reaction we are going to inhibit. That is why enzyme substrate and enzyme inhibitor interactions are slightly different from the drug receptor interactions. Now, this is drug DNA interactions because DNA is also one kind of a receptor. It receives molecules, and then the kind of mechanisms that are associated with drug DNA interactions are completely different from that of the drug receptor interactions. That is why we have categorized into this particular topic. Next is estimating the strength of these interactions. Each interaction leads to some specific amount of energy release. That much energy has to be supplied to break the existing interaction, which is already facilitated by our natural system. So now we go to the next topic of comparing and contrasting the interactions. Which drug is showing what type of interactions, how that particular interaction is helping in signal transduction or secondary messenger generation or the secondary messenger transduction or the cascade of events that are happening after the enzyme is inhibited or the drug receptor complex is formed. Many things are happening. But if I have a particular drug, a few things are happening with some amount of efficacy, and with another molecule, the efficacy is either increased or decreased. To compare that efficacy factor, we are going to compare and contrast the interactions, and then we are going to analyze them in terms of affinity. We analyze the affinity, we conclude in terms of efficacy. This is a game we are going to play. This is the research that we are going to do. This is the expertise we are going to develop. It all comes out after having some amount of expertise. When we compare and contrast, we can easily interpret that yes, a particular hydrogen bond is facilitating the interactions much better than the previous drug molecule. Like that, we can be able to conclude. That is what we are going to learn in the coming 2 to 3 weeks through this drug discovery hackathon training programs. Interpreting the results to design new species is art. It is not just science. Science always speak about facts and the interactions or energies and all those things. Whereas art of drug discovery is to interpret it in the efficient way. Which particular interaction is helping, it is not automatically coming from the science. Science tells us affinity. It does not tell about efficacy. So the art of drug discovery is between finding affinity versus the efficacy of a given compound with respect to its interaction with receptors or enzymes or the DNA or any other macromolecule. That's what we are going to learn. The art and science, we are going to learn both. The basics include art as well as the science of drug discovery. All the above depends on the basic chemical interactions. Let us first talk about the chemical interactions. We know some of them already. Let us see. Basic chemical interactions are in the form of covalent bonds, coordination bonds, ionic bonds, hydrogen bonds, van der Waals interactions, tautomeric processes, Like that, there are several chemical phenomena which we already learned. We have experienced also. We have experimented with reference to all of them. Now we want to revise this part. Why? Why do we need to revise covalent bond interactions? We know all of them. We know the basic definition of covalent bond. What is its current role? It will become clear to you in a few minutes, because whatever we understand in chemistry, we need to be able to translate it into drug discovery process. This is not a easy topic. I'm going to show a few examples how one can interpret and the importance of covalent bonds in the drug discovery concept. We will go to that right now. What is a covalent bond? We know it. It is bound due to the sharing of two electrons between any two atoms. That is being correct. We need to know how covalent bonds are important in the drug discovery chain. What is happening in the body whenever we supply any xenobiotic? That means any compound externally supplied. Of course, food is also a xenobiotic in one sense, but we are not talking about that. We are talking about the molecules that we designed and we are willing to supply to the human body with the help of a specific scientific process. That is what we are going to call as xenobiotic. Those compounds undergo metabolism. The metabolic reactions are all associated with covalent bond breaking and covalent bond making. And then prodrug to drug conversion. A prodrug is designed specifically to ensure that the drug is not degrading and drug is not acting at the wrong place. Drug has to act at the right place with the right affinity and with the right efficacy. For that purpose, we want to ensure that the drug is not getting affected with the help of many other enzymes. It is specifically attacking that particular enzyme for which it is designed. For that reason, sometimes we are interested in developing prodrugs. Sometimes the actual drug may be very, very reactive. It may get excreted very fast. So we don't want the excretion to go without showing its effect. So it should be absorbed, it should be distributed, it should be metabolized, and then it should show drug action, and then only we want it to get eliminated. For that purpose, we need the reactions happening on the drug should be slow. For that purpose also we develop prodrugs. When a prodrug is converted into drug, it involves either covalent bond breaking or covalent bond making. With this, we are going to the next topic, drug toxicity. On several occasions, drug toxicity is due to the fact that covalent bonds are formed where they are not supposed to be formed. Because of that reason, it is possible that we may experience drug toxicity. Now, this is one reason why we need covalent bonds. Let us see the example. Specific example is a very famous example. It was it is an anti-malarial drug. It is called proguanil. Proguanil gets converted into cycloguanil. What is the chemical reaction that is happening? How many bonds are broken? Yes, it involves the breaking of the CH bond. That is, CH activation is happening with the help of cytochromes. Then hydrogen radical gets abstracted. Then H radical, another H radical gets abstracted. That means actually H2 molecule is going out. However, we don't want to consider that it is H2 molecule, because when we look at the fundamentals, it becomes clear that cytochrome is removing H radical 1 and H radical 2. Two different H radicals are being removed. Then C-N bond formation happens as well as cyclization happens. The C-N bond formation and cyclization are together in this particular conversion of proguanil to cycloguanil. That is, a prodrug is getting converted into cyclic drug. I'm going to show it in the next slide. Then the cycloguanil gets protonated. That is also involving CH bond formation. After that, the cycloguanil is ready to inhibit PFDHFR, that is Plasmodium falciparum dihydrofolate reductase. Now let us see the strength of a covalent bond. It is approximately about 70 kilocalories per mole for a given CC bond. Now let us look at the example. The example is proguanil. I am showing the 2D structure of proguanil. You see that this is not a cyclic compound. It is having 5 nitrogens and 2 carbons, which are very elegantly arranged. It is called a biguanidine base. This is a functional group called biguanidine. This is an anti-malarial drug which was invented during World War II, very long time ago. Now, it is not a drug. It does not act as a drug. It does not show any inhibition of the PFDHFR. However, when proguanil is converted after protonation into cycloguanil, then what is happening? This CH bond, there is a, you see, isopropyl group is there. At this isopropyl group, there is a CH bond. That CH bond gets broken and H radical gets out, and then one of the NH bonds gets broken out, and then another radical is generated, and then cyclization happens. The bond formed is between this nitrogen and this carbon. This bond rotation, conformational change, all these things happen very, very elegantly. They happen within few seconds or few femtoseconds. This compound gets converted into this cycloguanil. Cycloguanil is a drug. Proguanil is not a drug. Both of them are highly basic. Proguanil gets protonated easily. Cycloguanil also gets protonated. Protonated cycloguanil is showing the drug action, whereas cycloguanil does not show drug action, and proguanil also does not show drug action, because this much positive charge on the cycloguanil is essential such that it can bind to the DHFR domain of the Plasmodium falciparum dihydrofolate reductase. Until and unless this positive charge is there, the binding between the enzyme and the cycloguanil molecule cannot take place efficiently. That is the reason why this is required. You can see that in this molecule, there is a nitrogen center with 2 lone pairs. 2 lone pairs at nitrogen are very rare. That is possible when nitrogen is having negative charge. However, this proguanil as well as protonated cycloguanil are positively charged species. Still, there are 2 lone pairs at that nitrogen. That is the beauty of this molecule. These 2 lone pairs, that means these 4 electrons are especially responsible for blocking the enzymatic action. That means electrons, which electron, where, how is blocking the reaction that is catalyzed by PFDHFR. That can be understood when we know what is the basics of drug action. That's what I'm going to emphasize. Let us look at this example. We all know about this molecule, Remdesivir. It is very famous today. It is its cost is being reduced. Our government of India is making special efforts such that this particular molecule is available to entire all the patients of the COVID-19. If this particular drug is available, but this is not a drug, this is a prodrug. This prodrug has to get converted into this particular drug, metabolite 4, M4. M4 is a drug, whereas Remdesivir is not a drug. Remdesivir is the one which goes to the chemist shop. In the chemist shop, you purchase Remdesivir, but once you supply this to the human body, it gets converted into M4, and M4 is a metabolite. This metabolite is the one which is inhibiting the enzymes. As a result, this particular conversion from Remdesivir to M4 is an essential process. Look at what is happening here. This particular group has to go away. First of all, with the help of esterase, there is an ester group here. The ester group gets broken and ester hydrolysis takes place. In this process, the carboxylic acid is generated at this center, either in the form of carboxylate ion or in the form of carboxylic acid. That means what is happening here? OC bond has to break first. This is the very first step in the prodrug to drug conversion. Next, look at what should happen. This molecule should undergo the loss of this particular phenoxy group. How the phenoxy group will go away? It will not go on its own. There is internal reaction that has to take place. What should happen? This particular oxygen, O minus, attacks at this phosphorus center, then this phenoxy anion goes out. Once that is gone, this turns out to be a cyclic system. That means a cyclic process is happening. OP bond is formed and PO bond is broken. So this particular covalent bond participation is happening here. After that, again, PO bond gets broken and M2 is generated. Once M2 is generated, it is ready for breaking the PN bond. See, first PO bond, first OC bond, then PO bond, then PN bond have to break, and then you will have a monophosphate available. This monophosphate is ready to get double phosphate and then take up another phosphate group. Inorganic phosphate gets attached with the help of ATP, and then this particular M4 is generated. This involves lots of covalent bonds. You can see that covalent bonds are very, very important in drug discovery. We need to understand which is being broken, which is being formed, which is a product, which is a drug, where is the toxicity, where is the safety, where is the covalent bond, how much energy is there with the covalent bond making and covalent bond breaking. These are all very important. Next, look at coordination bonds. Are they important? Yes, they are important for examination in +2. And we remember coordination bond is formed between two elements. One group provides the 2 electrons, another group accepts the 2 electrons. This is enough for +2 student. But now in drug discovery, where are the covalent bonds? Where are the coordination bonds? Metallo-enzymes are involved in coordination bonds. Cytochromes are associated with coordination bonds. Cytochromes are the ones which are causing oxidation and metabolism reactions. There is covalent bond formation. And then vitamin B12 is associated with covalent bond formation. And then zinc fingers are there. And likewise, there are several enzymes which are associated with covalent bond formation and breaking. Magnesium 2 plus is a part of many enzymes. It is at the catalytic center, and then there are coordination bonds formed due to this. Now, CC coordination bond is also possible, CN coordination bond also possible. These are all very important, and the strength of a coordination bond can be defined as about 25 kilocalories per mole. Look at this drug, cisplatin. This is an anti-cancer drug. You can see, if you see carefully, cisplatinum is 2 plus because there are 2 Cl minus are attached to it. This becomes 2 plus, but there is oxidation state of platinum is what is more important. Now there is, this is what we already appreciate that. Now, but there is a coordination bond between the nitrogen NH3 and platinum. Many people make a mistake. It probably is NH2 because how can there be 4 valency at nitrogen? This is what the people get confused because they think it is a covalent bond. But it is not covalent bond, it is a coordination bond coming from the 2 electrons in one lone pair from the nitrogen due to the donation of electrons from the nitrogen to the platinum center. Coordination bond is formed. Cisplatin, this anti-cancer drug, is very famous, very efficacious anti-cancer drug, which is involved in the interaction with the DNA. Look at cisplatin DNA complex is formed like this. What happens? Platinum chlorine bond is broken and another nitrogen platinum bond is formed. Now platinum is 2 plus. This is not oxidation state, this is a charge. In cisplatin, platinum is +2 state. Now we are talking about it has got +2 charge. Now it is able to accept electron density with the help of nitrogen donating N7 of guanine is donating to the platinum center, and then what happens? DNA gets bent. When DNA is bent, then the DNA does not get replicated. That means the DNA, this particular two guanines, when they are side by side, they produce sufficient distance between the two base pairs, and then the coordination bonds are formed, and because of these coordination bonds, DNA gets bent, and as a result, DNA replication is blocked. Thus, the anti-cancer activity is being realized. Now look at the coordination bonds in the drugs one more time. This compound is called metformin. It is a very famous compound. Most of you have seen these tablets probably because your uncle or your grandfather or your parents are probably using metformin as a daily dose. Morning one tablet, evening one tablet, as a diabetic drug. Now, almost all diabetic patients use this particular drug. But this drug is not active like this. This drug is inactive in this form as I am showing. Look at that. There are 1, 2, 3, 4, 5 nitrogens. There are 2 carbons. There are 2 more methyl groups. Then there is hydrogen bond here. All these things are useless, because this is not a drug. Drug is next to it. That is protonated metformin. It is This is protonated metformin. That is metformin hydrochloride. It is a salt. The salt is dissolvable. The salt is responsible for the tablet that we are preparing. The salt is the one which is traveling through the organic cation transporters into the body. It is getting influxed and effluxed with the help of the organic cation transporters. This cationic form is active form of the drug, whereas metformin neutral is not active. A small change. Look at this small change is bringing lots of change in the activity of the molecule. This protonated metformin is active drug, and that is being utilized morning and evening by many patients. Now, let us look at these particular interactions. Do we know? We have learned covalent bonds and coordination bonds. We are now going to hydrogen bonds. Do we know about hydrogen bonds? Yes, we all think that, yes, we know. What are we learning here? Yes, we have to learn about hydrogen bonds because they are ubiquitous. They are there everywhere. They are there between one macromolecule and another macromolecule, between one small molecule and another, between glucose and the enzymes which are metabolizing the glucose into pyruvate, and thus it is getting further converted into the small molecules like nucleic acids or even the nucleic acid bases and the amino acids, as well as many other small molecules are being generated, because that is where hydrogen bond is directly participating. And now, really, do we know about hydrogen bonding? Let us see. Is this a bond? First of all, we have to ask the question, what is hydrogen bond length? Do we know the length? It is very important parameter. Its average values can be stated to be between 1.8 to 2.8. Having done that, next question is, what is hydrogen bond angle? This is also a very important factor. Many textbooks, they do not write this factor. If and only if this hydrogen bond angle between one electronegative element the bonding hydrogen and another negative element, if that particular angle is less than 110 degrees, the chances of hydrogen bond becomes very, very weak. It is not hydrogen bond anymore. It is only electrostatic interaction. So hydrogen bond is a bond only when these two conditions are satisfied. Then what is the strength of hydrogen bond? It is between 4 to 7 kilocalories per mole for one hydrogen bond. If there are 3, it can be 21 kilocalories. If there are many, it can multiply into large values. Is this bond or an interaction? This is a very important question, because when we write hydrogen bond, the example, we write the delta H plus, delta plus on hydrogen and delta minus on oxygen. This is one positive charge, then another negative charge. Then is it not electrostatic interaction? Then if it is electrostatic interaction, where is the bond? Electrostatic interaction is an ionic bond. If it is an ionic bond, there is no bond path between these two. Is it a bond or not? This question we can ask, and we can also answer that. How do we confirm that there is a hydrogen bond is present here? Only hydrogen bond is sufficient? No, there is something called bond critical point we need to find along the bond path connecting the hydrogen and the electronegative element. If we can collect these two along the path, if there is a particular critical point called bond critical point, and if we can identify that, then we can confirm that there is hydrogen bond. So we are going to learn about it sometime later. But ionic bonds, they are strong, they are distance dependent. Salt bridges are involved in ionic bonds. There are many other varieties. Electrostatic interactions, attraction between positively charged center and negatively charged center. That's what we have seen. Delta H plus and O minus. Dipole-dipole interactions and NH3 plus and COO minus interactions. Very quickly, they get dampened if there is water surrounding the NH3 plus and surrounding the COO minus. If there is a lot of water, then the ionic interaction gets very completely get dampened. So these are some of the properties of ionic interactions. Now let us continue with our question. What is the importance of ionic interactions? That is because I already told you about organic cation transporters. These are the molecules, these are the proteins, or these are actually say proteins only. They are having the property to efflux and influx molecules. When organic cation transporters, when we talk about metformin is a cation, OCT3 is an enzyme which is helpful in the transport, and that means efflux and influx of the metformin drug. Now let us look at this hydrogen bond, how we define. This is the traditional definition without supplying the angle, but this is a distance is 1.8 to 2.8. That's what we are already told, angstrom units. Now this distance, we know that is not sufficient criteria. The criteria should also include the OHO angle should be between 110 and 180. If you see such interaction is not there between your drug and the enzyme or drug and the receptor, if such interaction with less than 110 is happening, then you can conclude that there is no hydrogen bond. If this particular condition is satisfied, then only you can say that there is hydrogen bond. Now look at this example. This is acetic acid. Inside the acetic acid, there is no hydrogen bond. That means there is no intramolecular hydrogen bond is not there. Intramolecular. Okay, here. Why? Because OHO angle is less than 90 degrees. There is no intramolecular hydrogen bond, but there is intermolecular hydrogen bond. This intermolecular hydrogen bond is between one molecule of acetic acid and another molecule of acetic acid. Two hydrogen bonds are formed, and both of them are satisfying this particular condition, that the bond length condition as well as the bond angle condition. Thus, there is an interaction called electrostatic interaction is also there, as well as bond is found, because there is a bond critical point between this hydrogen and this oxygen, and somewhere in the middle there is a point called bond critical point. If that bond critical point is found, there is a bond. If it is not found, there is no bond. That means in this particular monomer of the carboxylic acid, there is no bond critical point found. That means there is no intramolecular hydrogen bond, there is intermolecular hydrogen bond. This is all basics, but bond critical point is not a basic, which we are learning today. Now, how it manifests, let us see in this particular example. This particular compound is known as rosiglitazone. It is an anti-diabetic drug. This drug was in the market for many years. For about 12 years, it was in the market. Now it was withdrawn. But it is still a very good example to discuss basics of drug action. Look at this. This has got several pharmacophoric features. This is lipophilic side chain, essential. Then linker, which is flexible to undergo conformational change. It is essential. Heteroatom is essential for some kind of hydrogen bond formation. And this linker is also essential. Now this aromatic central fragment is going to provide some kind of stability when the rosiglitazone is interacting with the PPAR gamma. PPAR gamma has got several helices. Helix 3 of PPAR gamma interacts with the central aromatic fragment and thus provide stability to the interaction between enzyme, between the receptor and the drug. And now this acidic fragment is the one which is responsible for the actual drug action. This lipophilicity, this central aromatic ring, they are all essential, but they are not causing the actual reaction. Actual reaction is blocking the flexibility of PPAR gamma. Removing the flexibility of PPAR gamma is the reaction that is required. That is caused by this acidic head group. Let us see how this is important in hydrogen bonding. Look at this. This particular same drug, same features, everything is perfectly arranged in the same way, but now when it is interacting with PPAR gamma, here is a histidine. Another histidine is from here. There are hydrogen bonds between histidine. And now there is a tyrosine here. These are the very essential interactions. These three hydrogen bonds are responsible for the removing flexibility of PPAR gamma. If PPAR gamma flexibility is not there, cofactors or co-activators can be recruited. If co-activators are recruited, then the GLUT4 gets activated. GLUT4 transports glucose into the body. That means what is responsible? These hydrogen bonds are responsible for the uptake of glucose into the cell. If the glucose is outside the cell, it doesn't want to come into the cell. Until it comes into the cell, it cannot get metabolized. That means energy cannot be generated. Glucose becomes useless if it is not utilized. That is why in blood sugar patients, we notice that lots of sugar is found in the urine. Why? Glucose is not utilized. The sugars are not utilized. How they get utilized? Because of these three hydrogen bonds. Very, very clearly, the three hydrogen bonds, when they are formed, GLUT4 gets activated. GLUT4 helps in the uptake of the glucose into the cell, and once glucose enters into the cell, it is not in the urine anymore. It gets metabolized immediately, and lots of energy is generated. Now, what is responsible? These three hydrogen bonds. But now I have to do computer modeling. This is nature is already doing it. Suppose I am having a computer model which mimics these three hydrogen bonds. It is very clear to me. I have a very good computer model. But if these three are not there, I am showing in the next example. Remember this page and the next page. Both of them very clearly. Look at this. Here I am showing a tyrosine which is following a different conformation. Look at tyrosine, this is same tyrosine. It is having a conformation which is different. Now I am going back once. Look at this. It is forming hydrogen bond between tyrosine and hydrogen. There is hydrogen bond. In the next example, there is no hydrogen bond between tyrosine and the NH because the distance is too long. There is no interaction possible. Absolutely wrong conformation of the tyrosine. And if this is what is happening, that means my model is wrong. The computer-aided drug design will have utter failure if this is the way interaction is being shown. That means when I am paying attention to the computer-aided drug discovery, pharmacoinformatics-based drug discovery, I need to pay attention where are the electrons, where are the bonds, where are the interactions, whether they are properly formed or not properly formed. This is an example where hydrogen bond is not properly formed between the same drug and the same enzyme, and this is an interaction where hydrogen bond is properly formed. That means a drug discovery scientist, that means all of us, all the people who are participating in drug discovery hackathon have to pay attention whether I am showing the interactions in the proper way or I am showing in the interactions in the wrong way. That means my molecular docking, my QSAR, my pharmacophore mapping, my virtual screening, all of them will fail if this is the interaction that it is showing, my model is showing, and if it is not corrected, it is my mistake. That means it is the mistake of the scientist. That means we are all responsible to pay attention to the interactions which are happening. Now let us continue. Its importance has been noted. The model the bonds in an appropriate manner. That is very important for us. Estimate the energies we have to do, and then macromolecular structure should be prepared so as to maximize the interaction between the drug and the receptor. Then we can talk about the tautomerism and conformational change. We have just seen that small change in the conformation of tyrosine has caused lots of difference between the interaction between the drug and the receptor. Now, the next important interaction is the van der Waals interaction. Look at this. Due to instantaneous dipole, they are formed due to the instantaneous dipole. That means a dipole is created on the spot and immediately interaction happens and then disappears. The dipole disappears immediately. So that is why it is called instantaneous dipole. And it is a temporary dipole. It is due to small interactions, then but not easy to break. Until and unless you supply that small value, it will not break. Once it is formed, it will stay put. It wants to continue with the interaction. We have to make special effort to break. That is why it is called van der Waals interactions, very important in our drug discovery. We have to supply the energy of 1 to 2 kilocalories to break a van der Waals contact. Sometimes it is 0.5 also. Now, if that is the value, that means that much energy is released for each van der Waals contact. How many such contacts are there? If I have 10 contacts, I may have anywhere between 15 to 20 kilocalories of energy released between the drug and the receptor. Now, not very easy to break if it becomes many contacts are there and if it becomes more than 15 kilocalories per mole. Drug macromolecule interaction. Macromolecule interactions. Lipid bilayers, and then many things. Van der Waals interactions are there everywhere in the biomolecular environment. They are there during the drug discovery. We have to pay attention to van der Waals contacts. Many such contacts. We have to pay attention to each and every contact. For that, we can estimate the energy Lennard-Jones potential. That is energy which is proportional to distance. R is the distance. Sigma is a constant. It is proportional to 1 by r to the power of 12 minus 1 by r to the power of 6. That is what is Lennard-Jones potential. And then the original van der Waals contacts are due to the London dispersion. What happens when two neutral objects come close to each other? An instantaneous dipole is generated on object 1 and another instantaneous dipole is generated object 2. Because of that, partial negative charge, temporary negative charge is generated on one object and the complementary positive charge is generated on the other. There is contact developed, and that though the dipole gets disappeared, the contact remains there. That is the beauty of van der Waals contacts. We all have to understand, we all have to remember this very well. This is very, very important interaction. Now, van der Waals contacts in biology. All hydrophobic contacts are associated with van der Waals interactions. Cell walls are due to van der Waals interaction. Drug receptor interactions are due to van der Waals. Permeability of a drug through various components of the body, whether it is going through the blood-brain barrier or whether it is entering into the kidney or whether or not it is entering into your tissue, a drug is entering into your tissue or not. These are all due to the balance between hydrophobicity and hydrophilicity. That is defined by the number of van der Waals contacts. So transport of the drugs is due to van der Waals interactions. ADME properties originating from van der Waals interactions. A balance between hydrophobicity and hydrophilicity has to be understood by every drug discovery scientist. Now, likewise, now let us go into the theories. Occupation theory, rate theory, induced field theory, activation aggregation theory, macromolecular perturbation theory, two-state theory. These are all theories of drug action. These are all introduced by the drug discovery scientists over the past 60 years. But now we are going into computer-aided modeling. There we have the theories called docking theory or docking technology. Now we have the theory pharmacophore mapping theory and pharmacophore mapping technology, QSAR theory and QSAR technology. We have the virtual screening. We have many technologies like de novo design, or the molecular dynamics can also be used as technology. Quantum mechanics can also be used as a technology as far as drug discovery is concerned. So all of them are useful in recognizing agonists, partial agonists, and antagonists. That is very important. That is what we are going to do in the next. This is about the receptors. We are going to learn about the enzymes also in the next few classes. Drug receptor interactions are all, again, we are talking about this recognition of a drug by the macromolecule is drug receptor interaction. These are due to the receiving of the drug and then interactions and finding the cavity and binding in the cavity, releasing the energy and amplification of the signal. It is most important. If there is no amplification, the drug action is very small. Efficacy is small if there is no amplification. If interaction talks about the affinity, but amplification factor, it talks about the efficacy of your drug. And an example is insulin receptor. This is a figure. I don't have much time to spend here. This insulin binds in the middle of the extracellular domain, and then the action happens inside the cell. That means the receptor outside something happens, something else happens inside. Look at those disulfide bonds, sulfur-sulfur bonds. They pull the strings here and there. This pulling of the strings causes the phosphorylation happening at the ITRK domain of the internal domain of the IRTK domain of the insulin receptor. Then that is where the drug action is reflected. Though the insulin is binding outside the cell, drug action happens inside the cell. That is what is an example here. There are several theories of the enzymatic action, approximation, covalent catalysis, acid-base catalysis, electrostatic catalysis, transition state theory, slow tight binding, mechanism-based inhibitors. There are so many of them. I hope I will get one more chance to speak to you. I will explain all these things in my next class. For example, this is a GSK3 glycogen synthase kinase. You have to pay attention to the 3D structure of the enzyme, pay attention to the cavities. Thus, we may be able to prepare inhibitors like inhibition by metals, inhibition at the ATP binding site, inhibition at the catalytic trade, inhibition at the substrate binding domain, or irreversible inhibitors, peptide-like inhibitors, or inhibition by binding at the allosteric site. Like that, there are so many opportunities with reference to each of the enzyme. I am giving example of only one, that is GSK3, but such alternative examples of drug action are available, enzymatic action are available, inhibitor design is possible with reference to these many varieties of options with reference to all enzymes. Thus, this particular series of small interactions we learned today, which we already know, but we have relearned them because we want to pay attention to all of them in the coming 6 months during this drug discovery hackathon. Now I shall stop because already I have taken more time than what is allotted by the organizers. Now I think it is time to take questions. Organizers.
Thank you very much, sir, for your informative talk. I'm Dr. Himakshi Sharma, working as a postdoc under Dr. Sastri. So, sir, it is really very nice to heard from you about drug discovery. Almost you have covered all the important aspects of drug discovery, such as types of interactions, importance of coordination bond, covalent bond, and from prodrug to drug, etc. So, sir, we have received a lot of appreciation for your talk, and we have also received many questions. But due to time constraint, we have to take few questions. So, sir, may I start the questions around?
Sure.
Okay, yes. Sir. So, we have a first question asked by Anu Atrival. So, question is how this protonation proguanyl to cycloguanyl is occurring, which factor is enhancing it, and how other prodrugs get converted to drugs. What is the driving force?
The driving force is again enzymatic action. For example, proguanyl to cycloguanyl conversion happens with the help of cytochromes. Cytochromes are oxidizing enzymes. They have the oxidation means the removing hydrogen is also oxidation. So what they do is that the cytochromes have got a catalytic center. That catalytic center is radical in nature. That means it has got one unpaired electron. That unpaired electron containing catalytic center, when it is coming close to, or when drug is coming close to that particular catalytic center inside the cavity of the cytochrome, that hydrogen abstraction is facilitated. That CH bond breaking is facilitated. Otherwise, CH bond breaking requires about 60 kilocalories per mole. Because of the cytochrome, the energy required for that CH bond breaking forming the radical of the proguanyl is only about 10 to 12 kilocalories. Cytochrome is facilitating conversion of 60 kilocalories energy, a process which requires 60 kilocalories into another process which requires only 12 kilocalories. This is what is the purpose of all enzymes. So enzymatic action is responsible for the conversion of proguanyl to cycloguanyl. Yes. Next question, please.
Thank you very much, sir. So, second question is, it is asked by Agarapu Chandan. So, his question is, cassette lentation concept takes into account in which kind of prodrug?
there are so many varieties of mechanisms of conversion of product to drug. It is the specific question is related to only a few examples which I never came across. So I would not be able to give a right answer right at this point of time, but it is to realize that products are designed. When we are designing today during the drug discovery, we may not always design only the drugs. You have to keep in mind Prodrug design is also important component. You may have to think about that. That means you may have to consider two enzymatic actions. One may be the case of inhibition. One particular enzyme is the one which is being inhibited. Another enzyme is the one which is converting the prodrug into drug. That means you will have to design compounds which are not acting on only one enzyme. It has to be acting on another enzyme which first converts the prodrug into drug and then inhibits the enzyme of your desire. That is one thing we have to remember during our design process in the coming 6 months. Next.
Thank you, sir. So we will take another question. It is from Nivedita Rai. How we will determine whether the molecule have druggable property after some modifications, protonation / ionization, etc. because the molecule have been screened based on drug-like properties.
Yes, that is the very first step we will do. Drug-likeness properties are what we want to analyze. But when we perform molecular docking, we may have to dock not only the molecule that you designed, its tautomer also you have to dock, its ion also you have to dock, its cationic form, its anionic form, its derivatized form. We have to dock many varieties. At that time, sometimes the clues will appear. During the pharmacophore map generation, also clues appear. During the virtual screening, additional clues will come into picture. So drug discovery, once we started with Lipinski rule of 5, that part is over and we go to the end in a particular stream of activities. That is not sufficient. We have to revisit every step again and again so that it is possible that we might have missed a few points in the very initial stages. During the latter stage, we learned a few lessons. With the new lesson that we learned, we have to revisit our first step, second step. That means this is a cyclic process. The computer-aided drug discovery that we are going to do is a cyclic process. In this process, either the first step or the 10th step or the 20th step will give us the clues whether ionic state is important or tautomeric state is important. A particular conformational state is more important than another conformational state. Likewise, many clues we will get. That is why we will have to do this work repetitively, iteratively. We have to do the drug discovery process. Then things will become very clear. Next question.
Thank you, sir. So next question is from Surabhi Dikshit. Her question is, how do we decide that what type of linker in drug synthesis is needed?
Yes, we want linkers to provide flexibility to our drug. Very nice. But that linker should not be too much, because of the linker, the molecule should not be too much flexible. It should have sufficient flexibility and sufficient rigidity. Every drug, as we discussed, we need to have sufficient hydrophobicity and sufficient hydrophilicity. Exactly in the same way, we need to have sufficient flexibility to the molecule. At the same time, sufficient rigidity is also important. So if there is flexibility versus the rigidity balance we need to maintain, that means it is better to have compounds which have got linkers which are flexible with maximum of 2 bonds or sometimes 3 bonds. If we have a linker with 8 flexible bonds, then it may not be a very good idea. So we have to limit, initially, we have to limit the number of linker atoms. Then we dock another molecule with slightly improved linker size, maybe only one carbon linker, then 2 carbon linker, 3 carbon linker. Then during the docking process, we will come to know that, okay, a compound which has got 3 carbon linker is doing a better job than a compound which has got only 1 carbon linker. At the same time, we come to know that a compound which has got 7 or 8 carbon linker is not doing very good during my drug discovery docking experiment. That means I will have to drop that kind of thing. So best to start with 1 carbon linker, 2, 3, 4. Once we do this much in a homologous series, then we change the linker with electronegative element. One of the carbon is replaced with oxygen, or carbon replaced with sulfur, or maybe NR group. So likewise, the flexibility is modulated. If I have an NR group in the linker, for example, the flexibility of my linker unit is reduced depending on the size of the R group. If my R group is tertiary the flexibility is tremendously reduced. But if R group is hydrogen, there is sufficient flexibility, increased flexibility in my drug molecule. So we have to play with this. Using the molecular docking, we will be able to get very good clues in this context. Okay, next.
Thank you very much, sir. So, sir, we will leave the meeting now, and it is really appreciated that you have answered all the questions very nicely, and I hope your talk will help all the students to participate in Drug Discovery Hackathon. Thank you very much, sir.